So today's topic is food poisoning, a very common ailment, and every human being suffers once or twice, perhaps more than that, in their lifetime. So we are going to study this today. We will be confining our talk only about the bacteria responsible for food poisoning and the various fungi and their toxins which are responsible for food poisoning. Most of the cases of food poisoning are of bacterial etiology. So let us see how we define food poisoning. Food poisoning refers to an illness acquired through consumption of food or drink which is contaminated either with microorganisms or their products or toxins. The term bacterial food poisoning is restricted to acute gastroenteritis due to the presence of bacteria or their products, that is the toxins, in food. So in this way, we understand that food poisoning can be caused either by bacteria or fungi itself or by their toxins present in the food or drink. We can divide bacterial food poisoning in three types: infective type, toxic type, and infective toxic type of food poisoning. Now let us try to understand the first type, infective type of food poisoning. In infective type of food poisoning, there is multiplication of bacteria which occurs in the body, in vivo. With the food or drink, the person would ingest large number of microorganisms or bacteria, that which we call infective dose, and after ingestion of adequate amount of bacteria, the incubation period is usually 8 to 24 hours after the ingestion of food or drink. Typically, in most of the food poisoning cases, we find incubation period ranging from 6 to 8 to 24 hours. Now, the typical example of this type of food poisoning is Salmonella food poisoning. Very important organism, bacteria, belonging to the group Enterobacteriaceae, Salmonella, and a notorious organism causing food poisoning in men. The causative bacteria of infective type of food poisoning are Salmonella typhimurium, Salmonella enteritidis, Salmonella indiana, Salmonella Newport, and Salmonella Dublin. These are the types of Salmonella responsible for infective type of food poisoning. The commonest among these is Salmonella typhimurium and Salmonella enteritidis. Other bacteria responsible for infective type, these are Vibrio parahemolyticus. Vibrio parahemolyticus. Then we have Campylobacter jejuni, and then E. coli, the type, diarrheal type, these are enterotoxigenic E. coli and enterohemorrhagic E. coli. And also Shigella species, which is responsible for bacillary dysentery, as you know. Out of 4 species of Shigella, it is the Shigella dysentery type 1 and flexneri. These are commonly implicated in food poisoning. So the infective type of bacterial food poisoning is caused by Salmonella typhimurium, Vibrio parahemolyticus, Campylobacter jejuni, and E. coli, toxigenic E. coli and hemorrhagic E. coli, and also Shigella species. These are the commonest bacteria which cause infective type of food poisoning. Now, another type, that is what we call as toxic type. In toxic type, the disease usually follows ingestion of food with preformed toxins. That means the food is already contaminated by bacteria. They have grown, multiplied in the food and have produced toxins or their toxic products. Now, these toxic products, along with food, when ingested, this type of food poisoning we call toxic type of food poisoning. In this type, the incubation period is quite short, 2 to 6 hours. 2 to 6 hours. And a very good example of this type of food poisoning is staphylococcal food poisoning, very common. Staphylococcal food poisoning, which is, as you know, caused by the enterotoxins produced by Staphylococcus aureus. So, in contrast to infective type, where incubation period is longer considerably than what we see in toxic type. Toxic type, 2 to 6 hours. In infective type, it is 8 to 24 hours. A good example of toxic type of food poisoning is staphylococcal food poisoning, and the main toxin responsible is enterotoxin produced by Staphylococcus aureus. Other bacteria besides Staphylococcus aureus which cause toxic type of food poisoning are Bacillus cereus and Clostridium botulinum. Bacillus cereus and Clostridium botulinum. Bacillus cereus, again, this type of food poisoning is also common. So here, in nutshell, we can differentiate infective type and toxic type by incubation period. Very short incubation period, 2 to 6 hours in toxic type, considerably longer, 8 to 24 hours in infective type, and organisms responsible we have already talked about. Now the third type, which is mixed, infective toxic type, means with food, bacteria are also ingested in adequate numbers of their infective dose, and also they multiply in the body and produce toxins, plus they have already produced toxins in the food, so the person ingests bacteria along with preformed toxin. So this type of food poisoning is called infective toxic type. And a good example of this type of food poisoning is Clostridium perfringens. Incubation period of this type of food poisoning is 6 to 12 hours. Infective toxic type, good example is the etiologic agent Clostridium perfringens. Incubation period 6 to 12 hours. So in a nutshell, we can say in this type, infective toxic type, bacteria release toxins in the bowel. Now let us see those infectious agents which are commonly responsible for food poisoning. In this slide, in this table, we can see the common bacteria causing food poisoning, their incubation period, along with symptoms and the common food sources. You are able to see this slide clearly? Okay. Now, those organisms where incubation period is 1 to 6 hours. Short incubation period. These are Staphylococcus aureus. The symptoms produced by food poisoning of Staph aureus, nausea, vomiting, and diarrhea. And the common food sources are ham, poultry, potato, egg salad, mayonnaise, etc. Then we have Bacillus cereus. Again, symptoms are nausea, vomiting, diarrhea. The common food source is fried rice. Then Clostridium botulinum. Again, symptoms are common, nausea, vomiting, diarrhea. And the common food sources are canned food. So, three important and common bacteria causing food poisoning, where we have very short incubation period. Clinically, it is very important to remember this. 1 to 6 hours, Staphylococcus aureus, Bacillus cereus, and Clostridium botulinum. All three have common symptoms like nausea, vomiting, diarrhea. Now we come to those organisms which have a little longer incubation period, 8 to 16 hours. In this group, we have Clostridium perfringens and Bacillus cereus. Clostridium perfringens, the symptoms are abdominal cramps, diarrhea, and sometimes, but rarely, vomiting may be present. The common food sources of Clostridium perfringens food poisoning are beef, poultry, legumes, and gravies. Bacillus cereus, the symptoms are abdominal cramps, diarrhea, again, vomiting is very rare. And the common food sources are meats, vegetables, dried beans, and cereals. And the third group, where we have a longer incubation period, that is more than 16 hours, lasting 24 to 36 hours. The bacteria are Vibrio cholerae, which causes symptom watery diarrhea or rice water stool. The common food source is shellfish and water. Then we have enterotoxigenic E. coli, again produces watery diarrhea. The common food sources are salads, cheese, meat, and water. Then we have enterohemorrhagic E. coli. The common symptom is bloody diarrhea. In hemorrhagic E. coli infection, we have stool mixed with blood. The common food sources are ground beef, salami, raw milk, raw vegetables, and canned apple juice especially. Salmonella species, it produces inflammatory diarrhea, and food sources are beef and poultry, mainly beef and poultry, also eggs and other dairy products. Then we have Campylobacter jejuni causing inflammatory diarrhea. The food sources are poultry and raw milk. Shigella species responsible for dysentery called bacillary dysentery. Common food sources are potato or egg salad, lettuce, and raw vegetables. And then we have Vibrio parahemolyticus causing dysentery. And food sources commonly responsible are mollusks and crustaceans. So in this table, the infectious agents of food poisoning according to the incubation period, that is the symptoms which they produce after poisoning, 1 to 6 hours, 8 to 16 hours, and 16 hours. It's a good table. One would be benefited if you remember this according to the incubation period, because clinically it is clinically very relevant. Now we come to the mycotic food poisoning in which fungi are implicated. Fungi are responsible either by itself or by their toxic products. So the mycotic food poisoning, many fungi form poisonous substances, and ingestion of these fungi or their toxins with food, resulting in gastrointestinal diseases, which quite often involves liver and are seen to be carcinogenic. Mycotic poisoning is of two types. one which we call as Mycetism and another called MYCOTOXICOSES. Mycotic poisoning, two types, Mycetism and MYCOTOXICOSES. Now, let us see what this Mycetism stands for. In this type of poisoning, a fungus which is eaten itself causes toxic effects. Mycetism, the fungus which is eaten itself and then it produces toxic effects. It may cause gastrointestinal diseases. It may manifest as dermatitis and also even death. This type of mycetism or food poisoning we find in several varieties of poisonous mushrooms. These poisonous mushrooms, they have been identified as inedible. They are not eaten because they cause mycetism, where the fungus itself is eaten. Second type is MYCOTOXICOSES. Now, this condition, it results from ingestion of food or feed that contain mycotoxins. Means not fungi itself, but the product of fungi are present in the food or feed. So diseases that result from ingestion of food or feed that contains mycotoxins are known as mycotoxicoses. Mycotoxins, usually when these toxins produced by fungi, they are actually secondary metabolites. Mycotoxins are usually, these are secondary metabolites. Their effect is not dependent on fungal infection or viability. This is important to remember. Fungi produce this toxin or these toxic products in the food, and then there may not be another fungal infection. Fungi may not be present in the food. So mycotoxins are, one can say, the type of poisons. A variety of mycotoxins are produced by mushrooms, especially Amanita species. Remember this. Especially Amanita species of mushrooms is very notorious for producing mycotoxins. Another important thing to remember is that cooking has very little effect on the potency of these mycotoxins. So even if we cook thoroughly the food which has been contaminated with these mycotoxins, it can still cause disease, may cause severe or fatal damage to liver and kidney. Liver and kidney, most of the cases of mycotoxicosis, they fatally damage liver and kidneys. Now we will talk about some of the mycotoxins produced by fungi. Out of these mycotoxins, the commonest and very important one is known as aflatoxin. Aflatoxin. Some fungi produce mutagenic and carcinogenic compounds which are extremely toxic to experimental animals. Out of these mycotoxins, the most potent is aflatoxin. The most potent of these mycotoxins is aflatoxin. It is produced by or elaborated by Aspergillus flavus, Aspergillus species of fungi, and especially Aspergillus flavus and their related molds. These can contaminate peanuts, corn, grains, and other foods. Aflatoxin has been shown to cause hepatomas in ducklings and rats and have possible carcinogenic effect in humans, especially liver carcinoma. So this mycotoxin, aflatoxin, important thing to remember about this, the commonest source of this aflatoxin is Aspergillus species, especially Aspergillus flavus, and peanuts, corn, and other grains and foods. This toxin is present and has been seen to be carcinogenic in humans, especially to the liver. Now, besides aflatoxin, we have other mycotoxins which are also called ergot alkaloids. Ergot alkaloids. Poisoning by ergot alkaloids is also called ergotoxicosis, simply because it is due to the toxic alkaloids produced by a fungi called Claviceps purpurei. Claviceps purpurei, while it grows on fruiting heads of rye. Fungus Claviceps purpurei producing ergot alkaloids, especially found on the fruiting heads of rye. A condition known as St. Anthony's fire, a condition called St. Anthony's fire associated with consumption of bread and other bakery products made with contaminated rye. And this condition, St. Anthony's fire, there is red patches, burning sensation on the skin and mucous membrane. And that's why called St. Anthony's fire. These are oxytocic agents, means they simulate action of oxytocin. These ergot alkaloids produce effect that of oxytocin. So these are oxytocic agents promoting childbirth by increasing force and frequency of uterine contraction. Then other type of mycotoxicosis, human diseases caused by fusarium mycotoxins include elementary toxic A. leukemia. The mucous membrane lining of intestines become roughened. Toxic metabolites produced by fungi like psilocybin and psilocin are psychotropic agents used as recreation drugs along with LSD or semi-synthetic lysergic acid diethylamide. LSD, you must have heard the name of this semi-synthetic drug, a psychotropic drug. 60s and 70s, it became very famous because of the hippie culture, LSD. So these toxic metabolites produced by fungi like psilocybin and psilocin, they are also psychotropic agents. Now we come to the laboratory diagnosis of food poisoning. In food poisoning outbreaks, the samples which we take, these are vomitus, stool, or the suspected food materials. So these three are the ideal specimen for diagnostic purposes: vomitus, stool, and suspected food material. When we take food material, it should be homogenized or washed thoroughly in sterile diluents such as Ringer's solution before processing. First, we will go for microscopy. A wet mount preparation in saline is made to look for the presence of pus cells, RBCs, etc. Hanging drop preparation can be made of the liquid specimens to demonstrate motility, especially in case of Vibrio infections, especially Vibrio cholerae infection. We can see dilating type of motility. In Vibrio parahemolyticus, we can see motility, but not of dilating type. Gram stain is routinely not done because of the presence of normal flora. But in special cases, especially in Vibrio cholerae infection, comma-shaped bacilli may be seen. Now the bacterial culture. We can culture vomitus, we can culture food material and stool. They should be inoculated on appropriate media. In general, fecal specimens should be inoculated on media like enrichment broth, that is the liquid medium, selenite F broth, or alkaline peptone water. Also, we can use solid media, McConkie agar, and highly selective media like DCA, XLD. And for the culture, we take vomitous, food material, or stool as sample, and they should be, especially fecal specimens, should be inoculated on enrichment broth, selenite F broth, or alkaline peptone water. Also selective media like McConkie agar, and highly selective media like DCA, XLD, TCBS for identification of Salmonella species especially and Vibrio species. These media are commonly used for cases of food poisoning which are suspected of gram-negative bacilli. If we suspect Staph aureus, we can go for blood agar. Now, further identification is done by appropriate biochemical tests for enteric pathogens. Then we perform serotyping with a specific group of type-specific sera. And then sensitivity test is done to choose appropriate drug for treatment. Shigella, Salmonella, when we expect, then we go for XLD medium. When Vibrios, we go for Mansour's medium or TCBS medium. Now, this table shows features of common mycotoxins. These are aflatoxins, fumosins, fumonisins, trichothecins, ochratoxin, cyclopioxonic acid. Out of these, aflatoxin produced by fungal species Aspergillus. Sources are nuts and maize, and clinical condition hepatoma, hepatitis, Indian childhood cirrhosis, and Reye's syndrome. Then fumonisins produced by Fusarium species. The source is maize, and clinical condition is equine leukoencephalomalacia or porcine pulmonary edema, also carcinoma esophagus. Then we have ochratoxin, again produced by Aspergillus species, Aspergillus niger, also by Penicillium. The sources are cereals and bread, and it affects kidneys, nephropathies, and then some of the features of Mycetism. The mushrooms poisoning. Ergot alkaloid Claviceps purpurei is the fungal species which produces. Sources are rye flour and clinical condition St. Anthony's fire. Then we have coprin poisoning produced by fungal species coprin. Sources butter. Clinical conditions and taboos like reaction, vomiting, diarrhea, pain, abdomen, etc. Then we have muscarine produced by Inocybe fastigiata. Sources food, can be present in food, and the clinical condition simulates cholinergic effect. Then we have ibotenic acid or mucimol produced by fungal species Amanita pantherina. Sources edible mushrooms, Clinical condition, abdominal pain, vomiting, diarrhea. Then we have cyclopeptides produced by Amanita phalloides. Sources, edible mushrooms. So in case of common mycetism, what we have seen here that eating mushrooms is not safe sometimes. Though people like you, people must have eaten and perhaps may be very fond of mushrooms. Be careful when you eat mushrooms. This finishes our lecture on food poisoning, especially concentrating on bacterial type and the fungal type. Yes, any question regarding this topic? You can ask now.